once the action potential reaches the end of the cell at the synaptic bulb or synaptic terminal, there is going to be a synapse that it has to get across, a synaptic cleft, as you can see here. It's an actual space between the cells. So in order for the signal to get from one cell to the other, the electrical signal is going to be converted into a chemical signal in the form of these chemicals known as neurotransmitters. The question is now, what is the link between that electrical signal and the release of these neurotransmitters? As you can see at the top here, that there are these other channels called voltage-gated channels, voltage-gated calcium channels. So in response to the action potential, these voltage-gated calcium channels open up. Calcium is going to move into the cell, and the movement of the calcium into the cell now causes a process known as exocytosis. These neurotransmitters are originally stored in synaptic vesicles, and when the calcium moves in, it is going to trigger the fusion of that synaptic vesicle with the membrane, and therefore release its contents into the synaptic cleft. Once these neurotransmitters have diffused across the synaptic cleft, they are going to bind to specific receptors on the postsynaptic membrane. This again will be in the dendrites or the cell body of the postsynaptic cell. So here we have three different receptors. We have a receptor for a neurotransmitter that is going to open up a chemically gated channel. So this is the key thing here now, is that all the channels we've looked at so far were voltage gated. These are now chemically gated channels. So this is a chemically gated sodium channel. Here we have a chemically gated potassium channel. And here we have a chemically gated chloride channel. So depending on the receptor, okay, for the particular neurotransmitter, different channels may open up. If it is indeed a chemically gated sodium channel, sodium will move into the cell, making the inside of the cell less negative. Remembering it's sitting there at resting membrane potential, so it's going to make the inside less negative, and it's going to depolarize the cell. As we've discussed before, that is an excitatory postsynaptic potential, and through decremental conduction will go towards the area where the action potential will be triggered, which is the axon hillock. If the neurotransmitter bound to a receptor that was linked to a chemically gated chloride channel, chloride is going to move into the cell, in which case it will make the inside of the cell more negative, hyperpolarize the cell, and this IPSP again will travel towards the axon hillock. The other possibility here is that the channel is linked to a chemically gated, the receptor is linked to a chemically gated potassium channel, in which case potassium is going to leave the cell, taking its positive charge with it, causing again an hyperpolarization or an IPSP, and it too will travel towards the cell body, decreasing in amplitude, known as decremental conduction. Whether an action potential occurs here at the axon hillock is going to be due to the summation of all of these postsynaptic responses. And again, we're going to have two different types of summation. We can have spatial summation. This is due to different stimuli coming in at different locations at the same time. The other type of summation is temporal summation. And what happens here is, if depending on the frequency, if a cell is stimulated before it returns back, it will increase the amplitude of the postsynaptic response. That response then again degrades, but its amplitude will be higher at the axon hillock and may indeed lead to an action potential. So we have spatial and temporal summation. The end result is either we are going to generate an action potential, an all or none action potential at the axon hillock, okay, remembering it's going to be all, or we will have none, no action potential if the summation of all of these postsynaptic graded responses is sub-threshold. As long as this neurotransmitter is bound to the receptor, any of these neurotransmitters is bound, it's going to continue to open up these channels. The movement of ions will continue, and you will continue to generate graded responses, which could elicit an action potential at the axon hillock. So once we have the release of the transmitter, we then have to stop the release. It's almost like having a brake on a car. Okay, so what you have to do now is get rid of the transmitter, and there's 3 different ways of doing that. Okay, one mechanism is that there's going to be enzymes in the synapse which will bind to the particular neurotransmitter and degrade it, will break it down. Okay, enzymatic degradation. Another thing that can happen is that the neurotransmitter may actually just diffuse out of the synapse and be picked up by a glial cell. The third mechanism is the neurotransmitter may actually be taken back up into the presynaptic cell. Okay, so we have reuptake. So 3 mechanisms. We either have enzymatic degradation, we have diffusion, which will then be taken up by a glial cell, or we have reuptake into the presynaptic cell. So that pretty well ends the story, or it just starts the next story, which is going to be the same cycle again and again until you get to the final effector.